Cincinnati Early research results suggest scientists might be onto a way to preserve heart function after heart attacks, or for people with inherited heart defects called congenital cardiomyopathies. Researchers at the Cincinnati Children's Heart Institute report September 28 in Nature Communications that after simulating heart injury in laboratory mouse models, they stopped or slowed cardiac fibrosis, organ enlargement and preserved heart function by blocking a well-known molecular pathway. The WNT superscript 2 catenin signaling pathway is involved in several of the body's fundamental biological processes. After heart injury, however, WNT superscript 2 catenin signaling ramps up in cardiac fibroblast cells to cause fibrosis, scarring and harmful enlargement of the heart muscle, according to the researchers. Our findings provide new insights on what causes cardiac fibrosis, and they open the potential for finding new therapeutic approaches to fight it and preserve heart function, says Catherine Yutzi, Ph.D., lead study author and scientist in molecular cardiovascular biology. WNT superscript 2 catenin signaling is involved in many normal and disease processes, and it's tough to target therapeutically. But the idea that early targeting of fibrotic response in cardiac disease may improve muscle function and stop disease is an exciting new direction. Heart disease is one of the leading killers of people in the United States, according to the U.S. Centers for Disease Control. And a growing number of adults who are treated for congenital heart malformations as children, many who essentially had their hearts reconstructed, face lifelong potential health complications as a result. Adult congenital heart disease is of growing priority and area of scientific inquiry at Cincinnati Children's. Clinicians and researchers at the medical center are developing new clinical and research strategies for adults who are pediatric heart patients. This includes looking for what so far are elusive therapeutic strategies to repair scarred and poorly functioning heart tissues after cardiac injury or disease. Mice help show the way in the current study. Yutzi and her colleagues used a newly developed line of genetically bred laboratory mice that allowed them to determine how important WNT superscript 2 catenin signaling is in cardiac fibroblast cells. Fibroblasts are important to building the connective tissues and structural framework cells that help hold the body together. But in the context of heart disease, researchers are learning resident cardiac fibroblast cells cause a deadly mix of tissue fibrosis, scarring and diminished function. To simulate cardiac injury in the mice, researchers conducted a procedure called transaortic constriction to restrict blood flow through the heart. Some of the mice were bred so that following cardiac injury they did not express cardiac WNT superscript 2 catenin in fibroblasts. Control mice in the study continued to express WNT superscript 2 catenin following heart injury. The control mice exhibited extensive fibrosis, scarring and diminished heart function. Mice not expressing WNT superscript 2 catenin had diminished fibrosis and scarring, and the animal's heart function was preserved. Study authors stressed the laboratory research is still at an early stage, and it is too early to know to what extent it might apply to actual clinical treatment. But the findings of this study, and other papers involving different molecular pathways by other research teams, have intensified interest in understanding how inhibiting cardiac fibrosis improves muscle cell function and pathology. This will be a central question in future research heart fibrosis studies by Yutzi and her colleagues, along with looking for possible ways to target the WNT superscript 2 catenin signaling pathway for potential therapeutic benefit. Funding support for the study came from the National Heart, Lung and Blood Institute of the National Institutes of Health P01HL069779 and an American Heart Association fellowship to study co-authors Fuli Xiang, Ph.D., and Ming Fang, both members of Yutzi's laboratory team. Disclaimer: Autism UREC are not responsible for the accuracy of news releases posted to UREC by contributing institutions, or for the use of any information through the UREC system.